The plasma membrane.
Plasma membrane. Okay. So, have we done this thing? Glycocalyx, slime layer, capsule, all of them?
Yes, ma'am.
Yeah.
Okay, and mesosome, gramsole is also done. That's done. Prokaryotic ribosome, this was also done, right?
Yes, ma'am.
Yeah.
Okay, so today I think we have to start with the eukaryotic organisms, isn't it? The plasma membrane of eukaryotes, right?
Yes.
Okay, and what is the last thing you people have written? The plasma membrane of prokaryotes. Tell me.
No, ma'am, we wrote the beginning of prokaryotes.
All right, so switch on your video, students, and keep it switched on until I'm teaching. So talking about plasma membrane, it has another name, that is the cell membrane. Now, in the case of eukaryotes, it is the outermost membrane of the cell because there is no cell wall at all. So it is the limiting membrane of the cell, and the invention of also, but cell wall is not present in animals. The rest of the organisms like plants and fungus, they do have cell wall present. Now, microscope was invented in the year like 1950s, and because of the invention of microscope, we could now see the structure of the plasma membrane in a better manner. Now, the structure that, like, just like how there is a proper structure of DNA given, same way the structure of plasma membrane is also given, which was described by Singer and Nicholson in the year 1972. And they called it as the fluid mosaic model. And with the help of this model, they wanted to tell us about the fluid nature of the plasma membrane, that it's not a very rigid layer, rather it is a membrane that is soft and fluid. So what they said about this fluid mosaic model is that the nature of the plasma membrane is quasi-fluid. Quasi-fluid is semi-fluid nature because of the lipid. Okay, so there's a lipid bilayer that is there, and in that lipid bilayer, there is the protein, you can say, floating in it. Okay, so lipid bilayer, meaning two layers of lipids are there in which the proteins are embedded. Talking about the proteins, so further, the proteins are of two types, extrinsic, the proteins, they are of two types, extrinsic and they are intrinsic. Now, those proteins that can easily be extracted, we call it as extrinsic. So, for example, if this is the lipid bilayer, two layers of lipids are there. So, if the proteins are here, then you call it as the extrinsic protein, that it is easy to remove. But if the proteins are embedded in the towards the inside, then it will be difficult to extract it out. So those are your intrinsic proteins. Talking about the functions of the plasma membrane. So the most, because it is fluid in nature and because it has that property of, like allowing stuff to pass through it, because of that, it can support a lot of cellular functions like cell growth, secretion, endocytosis, in which the cell is capable of taking something from the outside towards the inside. And cell division also occurs because the plasma membrane is fluid in nature. It's not, you can say, rigid, so that the cell won't move easily. When we talk about endocytosis, so this one is the endo means inside, cytos or cyto refers to the cell. So taking stuff inside the cell, like you can say taking food particles, taking water, that is what we call as endocytosis. How does it happen? It happens by the invagination of the plasma membrane. Invagination of plasma membrane means that the plasma membrane folds around the thing and takes it inside. That is your endocytosis. The opposite of endocytosis is exocytosis, which you can also call as cell vomiting or effaging. So here, once the food has been digested, once the particle has been used, the excretory product or the product that is not useful, it will be thrown out of the cell or from the cytoplasm of the cell through the plasma membrane itself. So the stuff from the cell moving out, that process is exocytosis. And then from the outside moving in, that process is endocytosis. Is it clear?
Yes, ma'am.
Now, plasma membrane, because of its fluid nature, it has a property of transporting the materials across it. Transportation of materials across the plasma membrane, meaning, and this happens selectively, meaning some substances which are smaller in size, which carry less charge, they can easily be sent inside the plasma membrane. So that is what we call as cellular transportation. So cellular transportation is of two types, passive transport and active transport. Do you know anything about it? Active and passive transport?
Yes.
No. Tell me, what is the difference?
It's when
Say
the
whichever you want to
cell
say.
membrane it goes from lower concentration to higher concentration.
What is that, active or passive?
Active.
Lower
And
to
then
higher.
passive is when it goes from higher concentration.
And what do we mean to say when we say high to low or low to high or anything of that sort? See, passive transport is that kind of transport which will not require any kind of energy. Imagine two scenarios. Which one is going to be easier, one or two? Coming from the top of the mountain or going to the top of it?
Coming
Which one is it? Two is easy or one is?
two is easy.
Sure. You people are understanding my drawing.
Yes, ma'am.
So here in the second situation, we see that the person is trying to come down the mountain or the hill. And the first says that the person is trying to climb up. So obviously, whilst climbing up the mountain, there's going to be more energy that is going to be taken. And while coming down from the mountain, it's going to take less energy. That is from high area to low area, we require less energy. That particular movement happens across the concentration gradient. So same way, when we talk about the passive transportation, so it takes place along the concentration gradient. Along means from high to low. Okay, so that is why in passive transport, there is no requirement of energy. And it happens along the concentration gradient. The best example for which is diffusion. You people know that diffusion is the movement of substances from a region of their higher concentration to lower concentration through a semi-permeable membrane. Movement of water, like if you talk about the diffusion of water, then we'll call it as osmosis. So this also is high to low and requires no energy expenditure. But when we talk about the neutral solutes, neutral meaning they carry no charge, so they can also do passive transportation because there is no charge to hinder the process. So it would not require the use of energy and it will be transported along the concentration gradient. Remember two things, along the concentration gradient and against the concentration gradient. Along the concentration gradient means high to low. Against the concentration gradient means low to high. Now talking about the second kind of transportation is active transportation. Active transportation is uphill transportation, meaning that here energy will be utilized in the form of ATP, right? ATP breaking down and all of that will happen. And here the movement is from low to high, that is against the concentration gradient. So when we talk about the active transport or the uphill transport, So it is the movement of materials against the concentration gradient, meaning from low to high concentration. It requires energy in the form of ATP, that is adenosine triphosphate. So this talking about active transportation, it is an energy dependent process, and most of the charged molecules, bigger sized molecules, they are transported via active transport with the usage of energy. Is it clear, students? Any doubts?
No, mom.
Okay. Now talking about the cell wall. Now the cell wall is that rigid layer surrounding the plasma membrane of the cell. Now the cell wall is like present in the case of plants, it's present in bacteria and fungi. But always remember that the cell wall is absent in the case of animals. Now you people only tell me, plant cell wall is made up of?
Cellulose.
Very good, cellulose. Bacterial cell wall is made up of? Say.
Not sure.
Say. You were sounding P, right? Say the word. What is the word? You people are right. Everyone is doing. Say it.
Pepti
Ah.
dog.
Peptidoglycan. And what is the fungal cell wall made up of?
Also peptidoglycan?
No.
Oh,
Mom.
Ma'am, it's made up of ma'am. No, I don't make the cell.
It's all the components in it.
It starts with the C. Now tell me.
Chitin?
Chitin. Yes,
Okay.
always remember it by the spelling chitin. So you'll remember it. Understood? So plant cell wall is made up of cellulose. Bacterial cell wall is made up of peptidoglycan. And the fungal cell wall is made up of chitin. Now the plant cell wall is made up of cellulose, which is a polysaccharide. Polysaccharide means a complex sugar. And apart from the cellulose, the plant cell wall, it contains hemicellulose, pectin, proteins, different components are there, but the major component is the cellulose. Then we have the fungal cell wall, chitinous, because it is made up of chitin, algae, cellulose, galactins, mannans, these things are there. And remember, when we talk about the structure of the plant cell wall, so it has two regions present in it. First, there is like a primary cell wall present, and then there is a secondary cell wall. So the primary cell wall is the cell wall that is formed first, and then the secondary cell wall is formed. What happens, Anidhi? What's so funny? So the primary cell wall is the wall that is first formed. It is thin and it is, like you can say, expandable. Most of the young plant cells, they possess a proper primary cell wall. So primary cell wall is present in all the plants. But then when the plant matures, then a second layer of cell wall comes around and becomes the secondary cell wall. Okay, so that happens towards the inner side and it is thicker as compared to the primary cell wall. Now, in between the primary and the secondary cell wall, there's like a cementing glue substance present, that is middle lamella. So it keeps the cell layers or wall layers together. And this middle lamella is made up of magnesium and calcium pectate. This is very important. You can imagine by the number of stars I've put around it. Clear? Any doubts?
No, mom.
no, ma'am.
Okay, now in the cell, there are special structures that are responsible for providing connectivity in between one cell and another cell. And those structures are called as plasmodesmata. So these are the structures that come from the cell and they join two adjacent cells together and it's like a tube in between them. And they are helpful in the cell-to-cell communication. Talking about the all-in-all function of the cell wall. So first is that it provides rigidity to the cell. It provides a shape to the cell. It protects the cell from any kind of mechanical injury. It helps to keep the pathogens like bacteria, virus, fungus, all of that away. And it somewhat, to some extent, it doesn't, like, it allows the stuff to pass through it. It helps in cell-to-cell interaction. And always remember that you know that the cell can swell up, it can shrink down. So it doesn't let the cell to burst while swelling up, and it doesn't let the cell to collapse while shrinking. Not shrink, the cell can shrink, but it won't collapse. Why will the cell not collapse? Because of the presence of cell wall. Clear, students? Any doubts?
Yeah. No, ma'am.
Okay. All right, so after that, the next component of the eukaryotic cell is an endomembrane system. Now, endomembrane system, it is a system that consists of different types of cell organelles that are connected together. So when we talk about the various components of the endomembrane system, so there is endoplasmic reticulum, there's Golgi complex, there's lysosome and vacuole. All of these structures are connected to one another and they form the endomembrane system. Clear? So now we have to study about each of these cell organelles one by one. Any doubts, anything you people want me to repeat? No doubts?
No, ma'am.
Oh, wonderful. Now, see, talking about the endoplasmic reticulum, if you see that the nucleus continues as, like, one part of the nucleus continues in the form of this reticular network-like structure. This is what we call as the endoplasmic reticulum. It's like an extension from the nuclear membrane. So when we talk about the endoplasmic reticulum, so it is a network of tiny tubular structures that are scattered throughout the cytoplasm of the cell. It is made up of cisternae that are long, cylindrical, flattened structures. Then the tubules that are branched or unbranched structures, and vesicles are there, which are oval, vacuole-like structures. These three structures together make the endoplasmic reticulum. Now see, the space which is present inside the endoplasmic reticulum, like all this purple space that you see inside, this is the luminal space. Whereas anything outside that space will be called as extraluminal compartment. So luminal and extraluminal. Clear? Now, the endoplasmic reticulum, depending upon whether ribosomes are present on it or absent, they are of two types. Smooth endoplasmic reticulum, in which there are no ribosomes, so it appears very smooth and nice. So they are free of ribosomes. Whereas there is the rough endoplasmic reticulum, which has ribosome present on it. So when we talk about the smooth endoplasmic reticulum, see, this is how it looks like. It is not going to have any kind of ribosome present on it. Can you tell me what is the function of ribosome, students? Ribosomes.
I'm not sure, but they help in protein synthesis.
You are absolutely correct. They are responsible for protein synthesis. Now you see this one, the smooth endoplasmic reticulum marked over here. It has no circular dots on it. It's like a plain tubular structure. Whereas the ones with the yellow dots are the rough endoplasmic reticulum, which have the ribosomes present on them. So when we talk about the smooth endoplasmic reticulum, so they are free of ribosome. That is why under the microscope, they appear very, very smooth. Talking about what do they do? So the smooth endoplasmic reticulum, they synthesize and store the lipids, they store the steroids, they do the detoxification of drugs. And whenever the rough endoplasmic reticulum synthesizes something, it goes into the next cell organelle, passing through the smooth endoplasmic reticulum. So synthetic product of the rough endoplasmic reticulum, it passes on to the Golgi apparatus through the smooth endoplasmic reticulum. So SER is clear to you all?
Yeah.
Any doubts?
No, ma'am.
After that, there is the rough endoplasmic reticulum. Now, rough endoplasmic reticulum is the one that has ribosomes present on it. Okay, and ribosomes, as you know, are the protein factories. They're responsible for the synthesis of protein. Because of the presence of ribosome, they appear very, very rough. And remember that the rough endoplasmic reticulum is in continuous with the outer membrane of the nucleus. Talking about the functions of the rough endoplasmic reticulum, so because they have ribosome, so they will make proteins. And secondly, they will give rise to the smooth endoplasmic reticulum. So are the different functions clear, students?
Yeah, ma'am.
All right. Okay. So now tell me, what is the last thing you people were writing? You write it down. Last line you've written. Tell me that.
Ma'am, the beginning of prokaryotes.
Okay, you people write quickly. Okay. Characteristics are done.
Yes, ma'am, we wrote this much.
This much is done. Cell envelope and modification is done.
No, ma'am, we have to start from here.
All right.
Mom, we can scroll. You can scroll. Mom, done.
Both of you are done, students?
Yeah, ma'am.
Very good.
I'm done. I'm done. I'm done. I'm done.
Done, ma'am.
Both of you are done, students?
Yes, ma'am.
Oh. Done, students? Shall I scroll down? Students, shall I scroll down?
Yes, ma'am.
Okay. Aisha, you are also done?
Yeah.
Done, students. Done, students. Shall I scroll down?
Yeah, ma'am.